Just before we start, say thank you to all of you. Huge thank you to York Medical Society, Jane Mortimer at the back, who is the outgoing administrator. Jane's been a fabulous supporter of the foundation for 3 or 4 years now. And the opportunity for us to partner with such a high-profile, prestigious York-based professional organisation and to have the use of the facilities, we think is absolutely fabulous. Ladies and gentlemen, please, if we can just welcome Professor Thomas Hughes.
Well, what am I going to talk about? I'm going to talk about some work we're doing to develop better model systems to understand pancreatic cancer. The problem with the type of research we do, or one of the problems with the type of research we do in the lab, is that the cells we use and their behavior in the sort of dishes that we grow cells in, it just doesn't really look that much like pancreatic cancer. Now, don't get me wrong, these cells are from pancreatic cancer, they are pancreatic cancer cells, but it may be no surprise if you grow a human pancreatic cancer cell on a piece of plastic in a dish in a lab, it doesn't behave massively like pancreatic cancer. But I mean, what experiment can you do? I've got to do an experiment. I've got to use something as a system to understand. So it's the best we've got, but it's not perfect. And I mean, there are other systems available. So I think I've listed there that you can, if you want to, grow human cancer cells in mice, and you have to destroy their immune system, else they would just recognize the human cancer cell and destroy it. And that's a different system to work on. Is that better? Well, it's probably not better, it's different. But I mean, no surprise, it's a mouse, it's not a human, it's not going to be the same. And it's a human cell in a mouse, that's even weirder. It's not going to be the same. And maybe the final iteration of that is that you can grow mouse cancer cells in mice. But they're mice, and actually mice don't get pancreatic cancer, really, so you have to do some strange stuff to make that happen. So these are the model systems we have. We've got to do experiments using something, but they are not perfect. So that's a problem. What's the solution, or what is a solution? The top statement, it's kind of obvious, isn't it? We need to be growing cancer cells from patients in a more realistic environment rather than in a plastic dish, in something that vaguely resembles the environment that they'd normally be in, and then they're going to behave like pancreatic cancer cells should do, and therefore we can understand their biology and understand how to kill them, which is really what we want to do. So I'm going to tell you about 3 ways that we've done that, not in huge detail, so this isn't going to go on for very long, I hope. And they are, well, there they are. Patient-derived organoids is one technique to try and encourage pancreatic cancer cells from a patient to grow in a more realistic environment. I'm going to mention fibroblasts because I can't give a talk without mentioning fibroblasts. That's what Lewis is working on. And I'm also going to talk about some explant microfluidics cultures, which are maybe next level sort of model system if we can get the things to live for long enough. So I think there's just a slide on each, but first of all, I'm going to tell you where this comes from. So, I mean, clearly, these are actual cancer cells grown from patients, and to do that, it is surprisingly difficult, and you need a lot of different pieces to fit together nicely to have the opportunity to do that. And the first thing you need is patients. So we're recruiting pancreatic cancer patients at the Leeds Teaching Hospital, so in Leeds. And to the credit of these individuals who obviously have had a terrible diagnosis that they are probably not going to survive, they are all unbelievably supportive. And if you ask them if they'll contribute to research, I don't think we've actually had anyone say no yet. So the patients are fantastic. What else do you need? You need some sort of ethical framework to be allowed to do this. You can't just approach people and grab cells and do what you want. So we have an ethical framework to allow us to do that. That's based at the University of Leeds, where I have an appointment as well as York St John. You need people who actually do the interacting with patients. I don't do that. I'm not a medic. So it's the research nurses and the surgeons who actually interact with the patients and are ask them if they're willing to contribute to the research. And you need histopathologists because the surgeon cuts tumours out. That sample then goes to histopathology so that the histopathologist can choose the exact bit of that tumour which is most representative of tumour. If you and I look at this bloody piece of tissue, we would not be able to identify which bit of it is actually cancer. I'm not interested in the bits that aren't cancer, I'm interested in the bits that are cancer. Only the histopathologist has the skill to say, yeah, it's that bit, which to me looks the same as all the other bits, but apparently they can tell. And they give us a sample of that. And this needs to be fast, because if we don't do this literally within minutes, the tissue will have died and we won't grow anything successfully out of it. So this is the surgeon needs to provide us with fast tissue access. We literally run that down the corridor down to histopathology. They deal with it absolutely immediately, and we're back into my lab within probably 5 minutes. It could be 10. It needs to be that sort of arrangement. And then what do we actually do with it? It needs to go into a lab and be taken to some scientists to do some stuff. So we have separate individuals who've been doing the three things that I've talked about: organoids, fibroblasts, and explant microfluidic cultures. So those individuals are part of a gang who are making this all work. I've already highlighted that the patients are incredibly supportive of this, and Yazan, he's not here, but I will sing his praises. It's really the surgeon that has made this work for us by recruiting the patients and ensuring we get that piece of tissue quickly. There are challenges, actually, and this is, I hope there aren't any histopathologists in the room. No, they're incredibly supportive, but quite rightly, their focus is on having that piece of tissue in a state where they can do what they need to do in terms of looking at it, checking it was cancer, assessing whether the whole cancer has been removed or not. Their priority is to the patient in terms of using that to aid the patient's treatment. Their priority is not to give some random scientist a hunk of it. So that's been an interesting dynamic, I think, for all of us, but it does work. But it's they're kind of caught between me saying I want it and their clinical priorities. And actually, it sounds rather trivial. Just coordinating between all these teams is actually a real challenge. And one aspect of that challenge is that I, rightly, should not know patient names. I'm not a medic. I shouldn't get to know patient details. Therefore, it's actually very difficult for the medics to say, we've recruited a patient. Well, I can't tell you who it is. Well, how will I know how to get their piece of tissue? Well, we'll have to call them a code. And so the actual logistics of making all the different bits, some of whom can know patient details, some of whom can't, it's actually a real challenge. But we've got that. This does work. What is this? What are we actually doing? So patient-derived organoids. So the idea of an organoid is, I've told you we can grow cancer cells on plastic. They don't grow on top of each other. They grow in a sheet, I suppose, on a plastic dish. And that's a bit rubbish. Cancers are, of course, three-dimensional, really. So they should be growing on top of each other, but in a plastic dish they won't. So this is a method of making them do that. So we actually grow them in a piece of jelly, and then they populate the piece of jelly within 3 dimensions, and they arrange themselves because they recognize that they're in a 3-dimensional space. They actually start to arrange themselves in a orientation which is reminiscent of what they do in actual cancers. So just visually, if you look down a microscope and see how the cells are arranging themselves, it starts to look a bit like a piece of cancer. This is a much better model. So how do you do it? You get a hunk of tissue, you cut it up, you digest it so the cells are in suspension, so individual cells are floating around in a solution. Then you cast that into a couple of millimeters dome of a jelly-like substance, which is made of collagen mainly, which you might have heard of it. It's a component of skin. And you grow that, and you eventually, if you're lucky and you get all the soup that you have to include right, you manage to grow organoids. So these are organoids, or these are the jelly domes that I described. So the whole sort of circular thing, that's a jelly dome, and that's about 2 millimeters in diameter. And it starts looking like this, which is mostly just rubbish, sort of debris from the suspension of cells. But the picture to its right is what happens about 3 or 4 days later. And you can see that the cells have started to arrange themselves into these circles where they recognize that they're in an environment where they can move around and start doing some of those distinctive cancer-like behaviors. Moving around is a distinctive cancer-like behavior. And eventually they grow. So the next picture along is again those having grown and arranged themselves even more. Still quite a lot of debris in there. So the passage label means that what we've done is we've disaggregated that complete thing, washed it repeatedly, then put it back together, and that's got rid of some of the debris. And so by passage 1, you can see there's not really many random lumps in there. It looks like a whole load of these strange circles of cancer cells behaving as cancer cells should. Patient-derived organoids. So what are we going to do with this? Well, I mean, the idea, I suppose, is to try and assess whether this is a good way of understanding cancer cell behavior, and assess whether if we start throwing chemotherapies at these organoids, whether that accurately reproduces whether the individual cancer, the individual person should be treated with chemotherapy or not. Can we model that by looking to see whether their organoid is successfully killed by chemotherapy or not? Does that make sense? So that's the idea, but this is an important check on this whole process. So these are known as Manhattan plots. You'll see why maybe on the next one. And this is a way of looking at the DNA in the cancer cells and looking to see whether it looks reasonably normal or looks reasonably strange. Cancer cells tend to have strange DNA. But I suppose the idea here is that we've taken an original tumor and we've grown an organoid from it. So the key thing that I want to see is that the DNA in the original tumor and the DNA in the organoid, they should pretty much look the same, else my organoid isn't really very much like the original cancer. I want it to be like it. I want it to represent the original cancer. So that's what these are. If you look along the plot through the different colors, you can maybe see that there are little dots above and below. And if you look at those pattern of little dots, that's identifying bits of DNA that either have been deleted in the cancer or have been amplified, too many copies of that bit of DNA in the cancer. And that pattern should be the same in the original tumor and the organoid. And in fact, it is pretty much. So I'm going to say that this organoid is a really good representation of that individual's cancer and therefore is a really good model for understanding how that cancer should behave. Now it might become a bit clearer why these are called Manhattan plots. So if you look at the original tumor on that top one, you can see the sort of skyscrapers is the idea. These blocks resemble a sort of skyline of a city. This original tumor was a really strange looking genome. So the DNA in this original tumor is really badly messed up, which is not unusual for cancers. But the striking thing is the organoid looks absolutely nothing like it. So this organoid is rubbish. It really isn't very like the tumor. I don't quite know what we've grown out here. We seem to have grown out a very rare cancer cell, which might have been lurking in there. But most of the cancer cells had horrifically changed DNA, and we've not got that. So this is a really important check to check that your organoid actually looks like the thing it's meant to be representing. Patient-derived organoids. Have we done much with them yet? No, because we now have 9 in total from 9 patients, and we're just beginning to dip our toes into how to use these and how to do experiments with them. So patient-derived cancer-associated fibroblasts, many of you have heard me talk about this idea that fibroblasts protect cancer cells from chemotherapy. They do, but to do that experiment properly, we need fibroblasts that are from pancreatic cancer patients. So that's what we've done here. How do you get them? You get your piece of tumor, cut it up again, and actually, quite simply, put it in a dish with the right sort of soup around it, and the fibroblasts just grow out. And I can show you that here. hopefully you can see the black dot in the middle of this tissue culture dish is a piece of tumour. And you might be able to see there's a sort of halo of stringy things coming out of it. That's the fibroblasts grow out of that piece of tumour if you provide the right conditions for them to do that. Those are patient-derived cancer-associated fibroblasts. And if we really want to do experiments with them, we need to immortalize them, which means they will grow in lab conditions forever, ideally, although it seems that one culture has just died, so it clearly wasn't growing forever. So we've successfully done that as well. So I think we do now have the right model system, and quite a lot of them, but actually it is proving problematic. They don't grow forever as we would hope. So there's more to be done here. But the experiments that Lewis is doing is he's doing these co-cultures where he puts the fibroblasts in a dish, that's the green things, and cancer cells in a slightly separate dish, but they can talk to each other because they're in the same fluid, and tries to assess how the fibroblasts are protecting the cancer cells from chemotherapy if we throw chemotherapy into that strange sort of separated but together culture. Final one, microfluidics explant cultures. What does that even mean? So microfluidics is a strategy to try and grow cultures in tiny vessels and to pass fluid over them continually to try and keep them happy by giving them food that's passed over them. So we've 3D printed some small vessels, and we put a very small piece of tissue, glue it into the thing, and then attach it to pipes, and here it is, and feed that piece of tissue using syringes which are on a driver, so a machine that pushes the syringe continually, slowly. So this is an actual hunk of tissue. We've not grown individual cells out of it at all. So this contains the cancer cells, it contains the fibroblasts, it's probably got some immune cells in there, some lymphocytes, it's got all sorts of stuff. I mean, that really is the perfect model of a pancreatic cancer because it's a piece of pancreatic cancer. We've not done almost anything to it. But can we keep it alive? And the answer is yes, we can, because we can measure whether it's dying by looking in the efflux from the pipe. So the pipe in carries food, the pipe out carries stuff that the cells throw out. And if the cells are really unhappy, they throw out a molecule called LDH, and we can measure that in the efflux. These are data from 3 individual cancers that we've done this for. The purple one has worked the best, so that's the one at the bottom where there are low LDH levels initially. That means that the piece of tissue is pretty happy. And there's only a slight increase over the time course of 12 days. So that piece of tissue was pretty happy for 12 days. It's getting a bit ropey at the end, but that's plenty enough time for us to do some interesting experiments. Red piece of tissue was a bit ropey in the first place, and I think we understand why that is. That one took us more than half an hour to get from histopathology into the lab. So that's immediately telling us that time is really critical, and basically that piece of tissue was dying already. So I think this is a really powerful technique, and the thing we would really like to do with this, because it's fast, is we'd like to be treating these pieces of tissue with the chemotherapy agents that the patient could receive, and within 10 days we should be able to give an answer as to whether that chemotherapy agent has successfully killed cancer cells or not. And therefore, in principle, we should be able to tell the medics who are treating the patient, don't bother with this chemotherapy agent, use this one, because this one worked on the piece of their actual tissue that's pretty representative of their actual cancer, because we've not done much to it other than keep it alive. Early days, we've done 3 of these. I'll tell you that the sorts of things we're doing with these model systems, so this hasn't really been about science, it's been about the model systems that we can use to do science. The things that we're doing at the moment are, many of you will know about this project about fibroblasts and those protecting cancer cells from chemotherapy. That's Lewis's work, supported by the foundation. But we're also working on this idea of using photodynamic therapy to treat pancreatic cancer cells more effectively. So that will be an agent that you can add to the cells, does nothing, unless you illuminate it with a particular wavelength of light, and then it will kill the cancer cells. And we think there's a role for that intraoperatively. So the surgeon will remove what he thinks is, or he or she thinks is the tumor, and we can throw in some of this photodynamic therapy, light up the region, and that will hopefully kill any cancer cells just at the sort of edge of the hole that are lurking there. It should be able to take those out, is the idea. So we're working on that, and that will use the explant cultures and the organoid cultures. And we're also working on some inhibitors of a molecule called mucin, which we think has a role in pancreatic cancer. Yeah, I alluded to the fact that this is a surprisingly difficult thing to do, to access these samples. There's a whole massive team of people who have done it, really, and I'm talking about it even though I didn't do anything. So I should acknowledge all these people, and I should acknowledge some of the funders as well. So the Institute for Health and Care has funded this work specifically, as has Pancreatic Cancer UK, but the Three Bears have, of course, funded the fellowship. And I think that's all I wanted to say. So I will, of course, take any questions.